Hello, and welcome to this week's biology assignment, Modeling Mitosis. In this episode, our group will be discussing the stages of mitosis and modeling them using pop beads, magnets, plastic tubes, and string.
In this model, the blue beads represent the father chromosomes and the orange represent the mother chromosomes. Also, the magnet is the centromere, or the part of the chromosome that links the sister chromosomes together, and the plastic tubes are the centrioles, or cylindrical cell structures. The string represents the cell fibers around the chromosomes.
Another process that we won't show in this video, but is still important to know, is meiosis. Meiosis is a more specialized process that generates gametes, or egg or sperm cells, that combine to form a new individual. Before mitosis can even begin, interphase must take place. In interphase, the cell copies its DNA to prepare for mitosis. This is represented by the 7 blue strand, 7 orange strand, 10 blue strand, and 10 orange strands of chromosomes.
A duplicate of each chromosome, or a chromatid, is then formed. This is shown by joining each of the corresponding colors and lengths, 2 longer blue, 2 shorter blue, 2 longer orange, and 2 shorter orange chromosomes together to form a sister chromatid.
The plastic tube, or centriole, is placed at a 90-degree angle just outside of the nuclear membrane. The centrioles are cylindrical structures that later move in on the next step, prophase.
After interphase, you can now start the first step of mitosis, which is prophase. Prophase is when the chromosomes become visible as paired chromatids and the nuclear envelope disappears. But first, the chromatid, or a loose complex of DNA and protein molecules in the nucleus, must become condensed.
In prophase, the 2 centrioles are moved to the opposite sides of the cell. A string, which represents the cell fibers that hold the chromosomes together, was then tied around each individual chromosome. The end of the string was placed through the end of the plastic centriole to allow movement of the chromosomes without directly touching them. The nuclear membrane was then broken down, as shown by the removal of the nucleus. It is also important to note that the point of contact is the connecticore, which is on each sister chromatid. Also, the extensive network of microtubules make up the mitotic spindle, which pulls the chromosomes to opposite ends. In the next stage of cell division, metaphase, the chromosomes become attached to the spindle fibers, or in this case, the string. The imaginary plane that the chromosomes line up on is called the metaphase plate. At this point, the 2 sets of bead colors are attached to the string, and the model looks like this.
Anaphase is the stage in which the chromosomes move away from one another to opposite poles of the spindle. We then separated the chromosomes from their spindle fibers by pulling on the string. Telophase is the final stage in cell division. In telophase, the chromatids, or chromosomes, move to opposite ends of the cell and 2 nuclei are formed. The blue and orange strands are moved closer to the centrioles they were beginning to head to during anaphase. The nucleus paper was then added back because 2 nuclear membranes are being formed.
Cytokinesis is the physical process of cell division that divides the cytoplasm of a parental cell into 2 daughter cells. It occurs concurrently with mitosis and meiosis. The orange and blue strands are now completely formed into 2 separate membranes, which both include 2 blue and 2 orange chromosomes, as well as 1 pair of centrioles. A cell plate then forms midway between the 2 daughter nuclei. The cell cycle is now ready to begin again.